The Center of Excellence for Biosensors, Instrumentation, and Process Control, COBIK, is a privately founded research institution focusing on creating cutting-edge solutions and opportunities for high-tech entrepreneurship. COBIK was established in 2009 by 4 industrial partners and 4 Slovenian research institutions. Today, it employs 83 highly qualified researchers that work in 6 laboratories in control systems, instrumentation, bioengineering, biosensorics, and new materials. All of its highly specialized and successful laboratories are horizontally connected by the Open Innovation Systems Laboratory. Ever since its inception, COBIK's strategic orientation has been in the rapid development of solutions that are further enhanced immediately after the research phase in industry. In 2 years, our researchers have created 27 innovations, developed 16 prototypes, and launched 2 patents.
There are many other excellent achievements, the development of 2 unique educational programs, and a number of research activities that all promise results with the potential for transfer into industry by 2013. Our main advantage is based on research and development excellence and strong market insight combined with excellent financial engineering. This is what guarantees our success.
Since the fall of 2011, COBIK has been carrying out the Co-Invest project to establish a Southeast European regional platform for connecting newly established high-tech companies and professional investors. In the fall of 2012, the first International Investment Conference took place in order to link mostly Southeast European high-tech projects with international investors. Since 2009, COBIK has established worldwide research and business collaboration with internationally recognized institutions. In 2011, COBIK carried out intensive activities for international cooperation, including a link with a medtech cluster of 87 small companies from St. Petersburg, which is an opportunity for stronger integration with fast-growing markets in Eastern Europe and the BRIC countries. In 2012, COBIK established the first of 2 spin-offs, both in the field of biotechnology. The Advanced Sensor and Instrumentation Laboratory integrates expertise from analog, RF and microwave electronics, optics, computer science, and material science. There are 4 research projects operating inside the laboratory. The Optical Technology-Based Timing System project is working on improving timing and synchronization systems based on optical fiber connections and commercial high-performance telecom electro-optical components. There is a distinct need for such systems in geographically distributed scientific experiments such as particle accelerators, telescopes, and so on.
Modern technology favors transfer of such signals over optical fibers. This has many excellent properties, but also some drawbacks, like thermal elongation that needs to be compensated for in the greatest possible extent. Our ultimate goal is to achieve short and long-term stability in the range of 10 femtoseconds in the distances up to 10 kilometers.
An improved synchronization scheme with compensation for changes in the optical path will also be used in the Distributed Sampling Techniques project. The aim of this project is to simultaneously transfer both the reference and the user data signal over the same optical fiber connection in several directions and to synchronize events in the picosecond range. Within the Sampling with Embedded Supercomputing project, highly skilled experts in the FPGA and hard real-time programming with experts in high-speed digital electronics are investigating the performance of multi-core CPU and GPU units used as signal processing cores. The researchers at the laboratory typically perform basic research with proof of principle on pre-prototypes. Using their long-term experience and cooperation with industry, the later prototypes are designed so that they can be quickly upgraded into a product and put on the market. The quality of their work has also been proven through several papers published in research journals and presented at conferences, with registered innovations and through patents that have been filed and granted. Researchers at the Laboratory for Bioanalytics are using their interdisciplinary knowledge to develop cutting-edge solutions for the biotech industry and sustainable alternatives to antibiotics. Production and analytics of biological molecules are complex and require constant improvements to ensure safe, economic, and regulatory compliant production. The Laboratory for Bioanalytics has substantial expertise in developing upstream and downstream processes for biomolecules and viruses and offers scalable, GMP-compliant process development for proteins, plasmid DNA, live and inactivated viral vaccines, and virus-like particles. Analytics of biomolecules and viruses is integrated and is an essential part of any bioprocess. This is especially important to note because virus quantification and analytics could be a bottleneck in process development. With this burden in mind, we are developing biosensors applicable in production processes of viral vaccines, viral vectors, and bacteriophages, where fast, accurate, and reliable in-process methods are needed. To disseminate knowledge in biochemical engineering, we have developed an international, hands-on training program called Bio Downstream Technologies Course BTC.
The course is intended for researchers and engineers from the biotech industry engaged in upstream and downstream processing, for researchers dealing with the purification of biomolecules and viruses, and for PhD and postdoc students that want to gain theoretical and practical knowledge of downstream processing. The first such course was organized in 2011, and the second one is planned for 2013.
The second focus of the Laboratory for Bioanalytics is antibacterial research. Antibiotic-resistant bacteria are a serious global problem, and there is an urgent need for new antibiotics or alternative approaches to prevent and treat bacterial infections. One of the most promising alternatives is bacteriophages, viruses able to invade different bacterial species as their specific host. Research in the Laboratory for Bioanalytics is directed towards key issues of bacteriophage application, efficacy, and safety. As part of the project, we are isolating and characterizing bacteriophages or their enzymes against the most important foodborne pathogen bacteria of the genus Campylobacter salmonella and enterohemorrhagic Escherichia coli. The final goal of the project is bacteriophage-based products and tools that will enable effective control of food chain contamination and treatment of diseases in humans. The Bioinstrumentation Laboratory works in the field of bioanalytics. The laboratory facility is located in Ajdovština, in the Vipava Valley in southwest Slovenia, an hour's drive from Ljubljana. The laboratory has an interdisciplinary team of 8 employees with strong backgrounds in physics, chemistry, biochemistry, biology, microbiology, and electrical engineering. The research activity at the laboratory is primarily applied, with a strong interest in know-how transfer and the establishment of propulsive spin-off companies. Their goal is to develop a commercially viable and potent series of bioanalytical and preparative tools based on solid and rigorous design for biotechnology applications.
The core technology in our laboratory is based on robust synthetic biomimetics ligands called aptamers. Aptamers are short, single-stranded DNA and RNA molecules that have ability to bind with adaptive conformation to selected target with very high affinity. Aptamers are produced under in vitro conditions by using combinatorial chemistry, known as systematic evolution of ligands with exponential enrichment.
These synthetic ligands show numerous advantages over conventionally used protein ligands such as antibodies or enzymes. Their higher chemical and structural stability, feasibility for a wide range of targets, and economic production make them promising for numerous bioanalytical applications, especially where reliable and robust solutions are demanded. The laboratory is currently active in engineering aptamers selective for targets of interest, including protein, virus, and cell targets, and has competence for CellX on cellulose filter, magnetic beads, native electrophoresis, and viable cells. Applied research at the laboratory is focused on platform solutions designed for new bioanalytical and preparative tools. In this regard, the laboratory is developing in silico tools for the smart design of fluorescent aptamer bioreceptors, which are used in fast assays for conventional fluorimeters. Part of the research includes the development of highly sensitive dedicated fluorimeters. Research on aptamer preparative tools focuses on applications for fast isolation and pre-concentration of analytes. For this purpose, various chromatographic solutions based on particulate, magnetic, and monolith filtering media are being developed. The Laboratory for Control Systems is oriented toward research and deployment of new solutions in control systems. Its main focus is on 1. Time-critical networks where there is a need to achieve better synchronization and data transfer on standard network solutions. 2. Control systems for large experimental infrastructure. One such example is ITER, an experimental fusion project. And 3. Control systems for cancer therapy using particles. Control of medical accelerators based on medical safety standards.
Our most important conceptual improvements here at the Laboratory of Control System that we would like to share with other institutions and also continue to develop in the future include, first, real-time synchronized transfer over a standard Ethernet, basically aimed at improved redundancy and better reliability or determinism of data transfer while still maintaining standard and inexpensive solution. Second is a machine protection system, which is a fast and configurable interlock system with a specific feature to prevent, let's say, faults or interferences to harm our systems. And last is a control system framework based on FPGA technology, including signal processing, transmission protocols, and also easy connection to input-output modules. And here the main idea is to reduce the needed time to develop custom real-time applications that our customers need.
Our laboratory conducts pre-competitive research towards the development of advanced materials systems. The focus is on multi-scale modeling and simulation of new materials and new materials processing operations. The models are developed within the physics-based and artificial intelligence concepts and describe multi-physics situations such as coupled electromagnetic field and transport phenomena that range from microstructure evolution to the macroscopic system level. The focus is in development and characterization of new bulk nanostructured steels and aluminium alloys. The research is performed in collaboration with the University of Nova Gorica and Institute of Metals and Technology in Ljubljana, as well as with the industrial research teams of Store Steel and Impel companies.
The laboratory is involved in the modeling of industrial processes with artificial intelligence. The methods for a steel production process path are currently being tested. The final goal is estimation of influence of individual process parameters on final product properties. Different types of nanoparticles are added to steel and aluminum in order to improve the mechanical properties. Several related laboratory melting, casting, and rolling experiments are underway in order to achieve new nanostructured materials with the topmost quality. The related samples are mechanically tested and metallographically characterized. The Advanced Material Systems Laboratory researchers use two main analytical devices for material characterizations: an X-ray powder diffractometer and a photoluminescent spectrometer.
The powder X-ray diffractometer is the main instrument for characterization of the materials. We are using it to perform the phase identification and to characterize the crystallographic features of the materials. In some particular cases, we are also analyzing the particle size and the lattice strains of our systems. The other instrument is the fluorescence spectrometer, which is used to analyze the electronic structure of the materials. What we do is we pump up electrons to excited states, and then we analyze the characterization of emitted light during recombination.
The Laboratory for Open Innovation Systems consists of 15 researchers and is located at the Faculty of Economics at the University of Ljubljana. The lab functions horizontally as a bridge between cutting-edge, high-tech projects and the global market. We have 5 strategic projects. 1. Development of an internationally excellent business education program for managers in science called MBAR. 2. Research on open innovation models, international research collaboration in innovation, and advising for doctoral students. 3. Transfer of key R&D contents to various stakeholders in Slovenia and abroad, especially via the annual COBIK conference. 4. Monitoring of technological policy and annual measurement of innovativeness performance of Slovenian high-tech SMEs. 5. Coaching high-tech entrepreneurs and spin-offs from academia to get strategic or financial investors for entering the global market via the Co-Invest platform. At the moment, we are also launching a complementary strategic initiative that builds up the innovation environment in Slovenia called the Center for Innovation Research. The Center for Innovation Research is a complementary strategic initiative that builds up the innovation environment in Slovenia as it brings together, supports, and promotes research related to innovation. It pursues and propagates both basic and applied research on innovation.
The laboratory promotes a new paradigm of open and collaborative networks of scientists, entrepreneurs, investors and policymakers. It supports non-technological and technological disruptive innovative solutions. Our ambition is to be a well-known business platform in the region of more than 250 million people.